Hello everyone and welcome to the British Society of Geomorphology webinar. This is going to be recorded, this webinar, and it will be put on the website at a later date. It's a pleasure today to welcome Mark Smith. Mark is a Professor of Water Science and Health at the University of Leeds. His background is in hydrology and geomorphology, particularly using laser scanning and structure from motion photogrammetry to quantify topographic changes and map rough surfaces to understand their interactions with flow. His more conventional geomorphology research looks at large woody dams, natural flood management, and the impact of beaver dams. Mark's talk today covers what began as a side project over 15 years ago, looking at the relationship between hydrological processes and flooding in major African river systems and malaria transmission at both the catchment and continental scale. It's a pleasure to have you here today, Mark, and I'll hand over to you. Thank you.
Thanks very much for the intro, Hannah, and hello, everyone. So thanks for the invite to speak today, and I do appreciate that malaria is a slightly unusual topic for a geomorphology seminar series. But what I hope to persuade you of today is that both hydrology and geomorphology can play an important role in understanding malaria transmission and vector-borne diseases more generally. So there's loads of thank yous and sort of acknowledgements that I'd like to make as part of this. This is not all just me. It's a huge team of people, as I'm sure you can appreciate. And I'm going to save all that till the end. So I just wanted to acknowledge it up front, though, that this is a big collaborative effort with a number of partners. And given the unusual topic, I thought what I'd do is just give a bit of background to malaria first before jumping into two different scales of inquiry. The first being our work that we've done to look at continental scale models of malaria transmission, which we're trying to be the first to embed some hydrological thinking into that. And then second, zooming into the landscape scale, which is where the geomorphology comes in and things start to get an awful lot more complex. And throughout, I'm going to weave in a couple of projects that we've had funded in both Tanzania and Zambia over the last decade and a half. So, for understandable and obvious reasons, there's been a lot of focus on zoonotic diseases. So that's an infectious disease that can be transmitted to humans from animals. And particularly the role of land use change on promoting those spillover events. But as Steve Tooth's paper this year or last year highlighted, what about the broader impact of geomorphology on those spillover events? So what impact do they have on infectious diseases, sort of the general geomorphological process, rather than just focusing on land use change? And it's generally not a well-studied area. There are broader examples here that I'm not going to go into too much detail, but there are a number of things where a tectonic event, maybe an earthquake or a volcano, has had health implications and impact on disease transmission. So the classic example is in 1994, an earthquake in California, where it triggered an aerosolization of a fungus, which caused an outbreak of respiratory disease. More kind of speculatively and hypothetically, there's some suggestions that an ancient eruption of Stromboli might have caused a chain of events that resulted in the evolution of the smallpox pathogen, for example. But I think that is quite speculative and uncertain. But more concrete, we see links between geomorphology and disease transmission that go via standing water bodies in the landscape. The presence of stagnant water. So, for example, schistosomiasis, the flatworm that causes schistosomiasis, involves a stage within a freshwater snail, and its contamination of that water can infect humans and livestock. But by far and away, the biggest contributor to these things is this little critter in the middle there, the mosquito. Okay, so these are commonly implicated with the links between hydrology, geomorphology, and health, certainly vector-borne diseases. So, for example, dengue, Zika, but today I'm going to focus on malaria transmission. And that's because the malaria transmitting mosquitoes are quite fussy in terms of their breeding habitat. And so that makes geomorphology all the more important. Now, I want to try and keep it relatively light today, but of course, we need to just think about the whys here. And malaria is clearly a serious global problem. Every year, we still see around 600 100,000 malaria deaths each year from about 250 million recorded cases. Of course, the real numbers of those could be much higher. Two key points to note from the World Malaria Report in 2023 is that 95% of those cases were observed in Africa, mostly sub-Saharan Africa, and unfortunately the majority of those cases and deaths were in children under 5. And the reason for that is because if you can survive your first bout of malaria, then you inherit some sort of immunity moving forward. You gain some sort of immunity. So unfortunately, some of those tragic cases they're mostly under 5s. And we've made some great progress in malaria interventions and tackling malaria across the world. And these two graphs here show the reduction in the malaria cases in blue on the left and the mortality rate in purple on the right for the World Health Organization Africa region. So we can see that thankfully these lines are going down from 2000 onwards. But I want to just draw your attention that something around 2015, 2016 happens. These are PowerPoint lines. These aren't statistically fitted or anything, but I guess you can see my point there that both the decline of the incidence rate and the decline in the mortality rate have a point in 2016 where these gains aren't carried forward. So there's been some sort of stall in progress, and we're at a bit of a crossroads when it comes to tackling malaria at the minute. Now, malaria is an ancient human disease. It's been around for a very long time. So you wonder why we've not managed to get to the bottom of it earlier. And unfortunately, it is a very complex set of circumstances that lead to malaria. And a solution to that will require an understanding of quite local factors. And the classic quote from Hackett in 1937 is that, like chess, there's a few pieces at play here, but it can create an infinite variety of situations. So we need to understand quite a lot about a locality before we can target and tailor some interventions accordingly. And I want to make a really important note here is, in this talk, I'm going to be trumpeting hydrology and geomorphology for obvious reasons, but we're just one part of a big puzzle. And what I don't want to do is make it seem as though, we've got the answer to everything. The argument that I want to make is that, this is an interdisciplinary problem that requires an interdisciplinary team. And I would firmly make the case that hydrology and geomorphology should be well represented in those teams. But, without the ecology, without the epidemiology, without the understanding of demography, sociology, etc., we would only ever have part of the picture. So I'm not on about just tackling it as geomorphologists, but just that we can contribute to these teams. And you can see from my stack of things on the left-hand side there, that their climate tolerances of either the parasite or the vector can be quite variable. So there are 4 types of malaria parasite that affects humans, 4 or 5, but also the vector, the Anopheles mosquito that transmits the malaria parasites. There's lots of different species of those, and each of those will have different behaviors and different habitat preferences. So particularly the breeding site preferences that we'll come on to in a second. And where they're located relative to human populations. Okay, and then below that in blue, we're not touching any of these today, but I just wanted to acknowledge the importance of these things like immunity, about cultural practices and the economy and infrastructure that are all part of this puzzle. So while I don't wish to dwell on them today, I also don't want to ignore them or suggest they're not important. So moving on to think about how malaria is transmitted in the first place. We are the secondary hosts. It's Anopheles mosquitoes that are the definitive or primary hosts. And there's a lot of these Anopheles little critters. Okay, there's around 70 plus species of Anopheles mosquitoes are capable of transmitting malaria to humans. So that's out of about 500 Anopheles species. And as I've said, there's about 4 human parasite species, and they've all got different climatic quirks about them and different environmental tolerances. Today, because I'm going to focus on Africa, we're going to be focusing on Plasmodium falciparum, which is the more dangerous of the 4 types of human malaria. Now, trigger warning on the next bit of this slide. Okay, this is a quite scary graph of how malaria is transmitted. Fortunately, not to scale, but we can see it involves an interaction between an Anopheles mosquito and a human. So if we take the point where the mosquito bites the human, you can see that these tiny little tadpole filament versions of the parasite life cycle called sporozoites, they enter the human bloodstream and make their way to the liver, where they can stay for a while. And it depends on the particular species of the parasites to what it does there. But after waiting in the liver for a while, they'll burst out and emerge in this second form, these merozoites. And it's these little circular ones on that picture. These are the ones that attack your red blood cells and might be responsible for the clinical response to having this parasite. Eventually, these will become in sexualized forms, these gametocytes, and it's those that the mosquito will suck up into its proboscis when it bites a human. And that malaria cycle, the parasite cycle, then continues in the mosquito's stomach. So you get these oocysts that develop in the lining of the mosquito's stomach. These break through that lining, make their way to the salivary glands, and complete the malaria transmission cycle. A key point to take away from this slide, of course, is that the mosquito gets malaria from humans just in the same way that humans get malaria from the mosquitoes. The mosquitoes aren't born with malaria, they will contract it from biting an infected human. So let's take a puddle-centric view of how malaria is transmitted. So really focusing down on these environmental influences, and this has developed from a review paper that we did in about 2013, looking at these factors. So what you will see on this summary diagram quite a number of times is air temperature. So these different cycles will depend on air temperature. So how long it takes for the parasite to develop in the mosquito's stomach, and also how long it takes for the mosquito to develop eggs ready to lay, all depend on temperature. So generally, the warmer, the faster this happens. Now, all mosquitoes were born in a body of standing water, or in a body of surface water. So without the existence of a suitable breeding habitat that is this surface water, there is no malaria. So while it's only responsible for one part of this malaria cycle, if you remove it, it breaks the chain of transmission completely. You'll then notice that when the eggs are laid, we go through various different larval stages from left to right in this diagram as the larvae develop into a pupae before the adult mosquito will emerge. And there are various factors determining how long that takes. So it's an absolute minimum, it will take 10 days to develop from an egg to an adult, and that's under optimum conditions. And if it's a little bit cooler, it might take a little bit longer. So yeah, that's really where the geomorphology and hydrology come in. But a few other points to make. Survivability also depending on humidity and air temperature. Because these mosquitoes aren't born with malaria, they have to have bit a human with malaria, and they only bite to take energy to lay eggs. Okay, it's not part of their feeding, it's really part of the reproduction cycle of the mosquito that they take a human blood meal. And only the female mosquitoes will do this. Now, they have to have found an infected human and taken a blood meal before the malaria parasite will develop in the mosquito. And then it's how many times around that cycle that mosquito goes as to how much malaria transmission takes place. Okay, so it's not just the female mosquitoes that you've got to be worried about, it's the grannies. The grannies that have been around for a long time, they're the ones that are most likely to have the malaria parasite already, and so they're the ones you don't want to get bitten by. The second point is dispersion. Okay, the wind and humidity will determine how far these mosquitoes will travel, and it's usually several kilometers. So if you think about it from a hydrological perspective, then our challenge is to identify an area where there will be a body of surface water that will last long enough, minimum 10 days, for the egg to become an adult, and we need to know to within a couple of kilometers where those are likely to be. So there's not much point being too much more accurate than, say, a kilometer resolution here, but we're nowhere near that yet. And then just a bit more background before we jump into the details. It's worth thinking about where malaria is in the world today. So this is all different types of malaria. And there's this excellent Malaria Atlas project, which maps where malaria is. But the problem is that where malaria is today doesn't necessarily correspond with the areas that are environmentally suitable for malaria transmission. And that's because we have made this great progress over the last few decades in shrinking that global malaria map, particularly around the DDT-driven malaria elimination campaign. So this was the global eradication strategy, tellingly did not include sub-Saharan Africa, but that made huge inroads throughout the rest of the world to try and reduce the malaria geographical range. But if we're to understand and predict changes in that, we need to think about the climatic or environmental envelope of suitability for malaria. And so for that, we need a more process-based approach to account for the influence of climate on both the parasite and the vector. And that's important because, we've seen a lot of the biggest advances in malaria eradication at the fringes of where is suitable. But as climate changes, actually, the length of the transmission season in some of those areas might go up. And certainly where that is higher, then it becomes harder to try and break that malaria cycle. More useful for us are these pre-industrial sketch maps of where malaria used to be, the pre-intervention, okay, before we did any of this. And there's this classic one by Lysenko and Semanchko in 1968, where it's basically a sketch, but it's as good as we've got of where malaria was around 1900. It's worth pointing out, zooming into the UK and this other map here, the dark shading is where malaria transmission took place. And so, yeah, we had malaria in the UK. You can see it goes all the way up to York, more or less, in the north. And this was not the falciparum malaria of Africa, but it was Vivax malaria. And, we drained a lot of these areas, but we were always on the fringes of malaria suitability, certainly back then. So it was a relatively easy task to eradicate the UK from malaria. There were other things going on. It was quite a zoophilic vector, so it quite liked to bite animals, not so much humans. And as well as that drainage came an improvement in living conditions more generally, and a shift from cattle-based agriculture to arable agriculture, which of course had an impact on the malaria cycle as well. So it wasn't just the drainage that was responsible. And the final bit of background, this is what it's all about, okay? It's about understanding the best malaria intervention to implement in a particular place, and also to prioritize places, where can an intervention have the most impact? Can we focus those somewhere? So you can see there's a range of things here. Insecticide treated bed nets are a very, very big part of this. Also spraying indoors, drugs, larval source control, so actually a larvicide in the puddles themselves, and then education, improving access to healthcare, and then possibly moving into the future, this malaria vaccines that we're starting to hear about. We've seen the flatlining in the progress against malaria, and the World Health Organization says that the way through this is to increasingly target and tailor our interventions based on the local epidemiological circumstances. And so this needs to be informed by epidemiology and ecology, but I would also argue hydrology and geomorphology are a part of that. And this is so important because the phrase that you hear is there's limited financing relative to the needs. So even where you have a useful intervention, you have to get the most bang for your buck. You have to target that. It can't just be blanket yet. So let's actually get into some examples now. I'm going to begin with our continental scale work before we zoom in into a bit more detail. So when I first started this 15, 16 years ago, I was kind of surprised to find that when we looked at climatic controls and talked about surface water, it was very much a black box. They said, okay, if it rains enough in a month, we're just going to assume that means enough standing water for malaria. Okay, there's zero hydrology. It was bypassed altogether. And when you looked at the literature into the rainfall threshold, the monthly rainfall threshold that you need for suitable malaria transmission, people had measured this in different places, and that the classic number that you see is 80 millimeters a month from the Mara model. And you also see 60 millimeters a month as well, with an 80 millimeter catalyst month. Those are the sorts of the typical figures that we see. But because it's not really about rainfall, it's about surface water, a wider scan of the literature will find actually an incredible range of different thresholds that exist, because these will have been measured in different areas with different hydrological processes going on, different evaporation rates, different infiltration rates. I know one of the sites that the Mara model was developed at, you'll see it later, in Tanzania. And the reason it's got loads of malaria is there's this whopping great big continental scale river that goes through that site that's responsible for a lot of the water bodies that are being formed. And it's got nothing to do with the local rainfall. This water's travelled from hundreds of kilometres, if not 1,000 kilometres away. And there's not been much work in understanding this rainfall threshold. There's been a lot of work looking at temperature, which I've already shown is really important for the malaria transmission cycle. And I don't want to ignore that here, that the thermal response curves are generally quite well known. There's still some uncertainties, but they're generally quite well known. And I guess it's not the topic for today, is all I can say on that. But we can apply those thermal curves alongside this surface water representation to try and get an idea of the environmental suitability for malaria. And you typically get a map that looks like this. Okay, so here we've got Africa for the period of 1971 to 2005. And a rainfall-driven model would give you an output that looked like this, with the response, with the map that we're looking at here, mapping season length. So the number of months suitable, the number of continuous months suitable for malaria transmission. So where is it warm enough? Where is it wet enough, essentially? And so a typical map would look like this. Now, in our first attempt at this, 2020, we coupled the climate data with list floods, not the floodplain list flood, but the hydrological list flood, to try and start to represent some of these surface processes. I don't want to dwell too much on the detail here because there's an updated version coming in a few slides. We only did this with RCP 8.5, okay, and with 7 climate models using list flood. But you can see a clear difference in outputs. Rivers emerge as important foci for malaria transmission year round. But we see in a map of how that might change to 2100. That's the rainfall-driven map. That's the hydrological-driven map underneath. And we see areas of both increasing and decreasing suitability for malaria in the future. I'm not going to go into any more detail because you'll see this again in a couple of slides. But one thing to say is that there was a big sensitivity to the rainfall threshold used in these rainfall-driven approaches, and yet a big uncertainty as to what that should be, looking at the different models in the literature. Now, validating these outputs is really difficult because of these interventions having reduced the malaria map. So the best we can really do is go back to 1900, to this pre-intervention state, and look at this Lysenko map, which you can see on the left here for Africa. And in our follow-up paper, we had an ensemble of 4 climate models with 7 hydrological models, and all combinations of those using the ICIMIP website data. And what we did was we weighted that ensemble of climate and hydrological models based on its ability to match different levels of endemicity, of transmission intensity across Africa. So the maps on the right are just for any malaria. There's actually not much difference between the two on those, but it became more apparent at the different levels of endemicity. When you do a quantitative comparison of rainfall, PR, precipitation, or a discharge-based approach on the right, actually, they're more or less identical. The hydro approach is slightly better, but not by a meaningful amount. But what you notice is that both models will over-predict in South Africa. So the green colour there is where we model it, but don't map it. Our runoff-based approach, our hydrological approach, suggests that the Orange River corridor would be suitable for malaria. That's not in the sketch map, but we do note that there are records of there being malaria there in the early 1900s. So again, we realise that our sketch map is not a perfect point for validation. We can also see under prediction in the Horn of Africa as well on both sets of models. One other thing to note here is that we've developed things a little bit further in this example. We're also using a ratio of rainfall to potential evaporation in there to catch those more locally driven puddles that wouldn't be in the hydrological setup. So this was the recent output at this continental scale. And if you compare the rainfall on the left with the hydrology predictions on the right, you can see that when you include hydrology, you get a much more complex pattern of hydroclimatic malaria suitability that's probably more realistic. It inevitably highlights river corridors as year-round foci of transmission. But we should note here that water flowing in these large rivers is not always necessarily suitable larval habitat. If it's fast flowing, it might not be used, but it's those grassy areas around the river banks, that poached ground that when the river drops in level is often a really suitable malaria larval habitat. Can be highly productive habitat. But when we look at the differences, you know, the one that jumps out first, right, is the Nile corridor, okay, is being suitable for malaria transmission, but it doesn't come up in the rainfall-driven models. Now, interventions have actually cleared this area of malaria since the 1990s, but we know it is still suitable. It's just that we've managed to remove it from the local transmission for the time being. Similarly, the Niger and Senegal rivers, Juba and Shabelle rivers in Somalia are all coming up as climatically suitable. And the obvious question from here is then, you know, how might this impact projected future changes? Well, all models agree that malaria suitability in Africa is actually going to go down. Okay, it declines to 2100. So by this is the most extreme RCP scenario. You do all of them this time, but we can see that by 2075, we lose about 500,000 kilometers squared of area that's suitable for malaria transmission, going to just over 1 million kilometers squared by 2100. So that might seem like a really good thing, but bear in mind, you know, if it's too hot or too dry for malaria, then you've got a whole host of other problems here. So I don't want to sell that as a good news story. Moreover, you know, the total area isn't really what we're interested in here. We're interested in where shifts. So if there is a reduction, but actually a big shift from one place to another, that will shift the locations of the people that are vulnerable, and there's issues with not having developed immunity to malaria in that time. So we need to think about exactly where those areas are. I mean, one example is sort of, if you can take these two maps on the right, sorry, not those circles, you can see in Ethiopia there's a patch of red, and that's an increasing malaria suitability, and that's thermally driven. That's why it's common to both models. That's the Ethiopian highlands getting warm enough for malaria transmission. But around the edges, we can see these big areas of decreasing malaria suitability, particularly in West Africa. Now, other studies have identified this. It's getting too hot, it's becoming too dry, but we're seeing it in our models much more pronounced than seen in other models. Overall, we see these shifts to be less extensive, but more concentrated. So where there is a shift in malaria suitability, it can be up to about 4 months of a shift. So this can have a big impact on the local transmission cycle, going both ways as well. So we can also see another area of decreasing suitability in the south, sort of Mozambique, Botswana, but also again, South Africa is coming up. Certainly the Orange River corridor is becoming more suitable for malaria. Now, also important is the sensitivity of that shift to the choice of emissions scenario. And in the bottom left here, this graph looks at the areas that are increasing suitability, decreasing suitability, and the net. So red increasing, blue decreasing, and then the green represents the net change. We can see again it's the high emissions scenarios that show the biggest swings, both in increasing areas and decreasing areas. So these are things to consider. Also, we've set a decreasing area, which is good news, but remember, our key thing here. It's actually human populations. And as we all know, the rivers are really important to be picking up, as we do in our model, because that's where the people are. Okay. And the projected growth in the human population in Africa to 2100 means that despite the shrinking area, there's an increase in the overall number of people projected to be living in malaria suitable areas to that time. We'd expect it to increase by about 80 million people to 2100. So again, including hydrology actually picks these things up, and we show that the number of people estimated to live in a malaria endemic region with a transmission season of over 9 months to 2100 would actually be 4 times greater when you use hydrology as part of your predictions rather than just using rainfall. So now it's time to introduce the geomorphology and zoom in for a more complex story here. It won't be a surprise to many of you, but not all puddles are the same. Okay, literature will talk about things like swamps, river fringes, puddles, drains, burrow pits, where you've sort of dug in for building material and then you've left a pit left over. These are always vaguely described and certainly not described in hydrological or geomorphological terms. So, you know, there's a very low bar here as a starting point. And it's important that different surface water bodies behave differently, because also mosquitoes, the Anopheles mosquito species, are all different. They have different feeding behaviors. Some will bite you during the day, some will bite you at night, some will bite you inside, some will bite you outside. Some prefer cattle, some prefer humans. They also have different breeding site preferences, you know, where in the landscape they'll breed, and have different climatic tolerances as well. Now, there's a diversity of species. What we're seeing here is a map of the dominant vector, but there's secondary vectors within any landscape as well. So it's not as simple as that map would suggest. Across Africa, though, we see that the main culprits that come up time and time again are Anopheles gambi, Anopheles arabiensis, and Anopheles fenestus. So gambi are, they love a small sunlit water body. It might be transient and a peri-urban setting, and it likes to bite humans, very anthropophilic when it comes to biting. Whereas arabiensis prefers cattle, to be honest. It would very zoophilic, but it likes to colonize these recently flooded areas, so sort of open sunlit pools on a floodplain after the flood waters passed. And then the third one to mention, Anopheles fenestus, this again likes to bite a human, very much so, quite an efficient vector there, but it prefers shaded, more persistent habitats, okay, so more year-round water bodies rather than these little pools. Long been recognized the importance of geomorphology in understanding where malaria transmission will take place. And in that 2013 review paper, you know, this is a figure from there that was taken from 1916, ultimately, this map of malaria transmission across parts of India and Bangladesh. And a few things pop out from this map. The first is that close to the river, malaria decreases, and that's because of the flushing flows. That the high flows of the river mean that it's not really suitable habitat there. The bigger pattern, though, is that you've got the more active delta in the east and the drying delta in the west. There's not much malaria in the active delta, and it turns out that the local vector there, Anopheles philippiensis, doesn't like a high water table. Okay, so that depends on the breeding preferences of the vector there as to where will be suitable. And it's that older drying delta further west that had much higher rates of malaria transmission. And what we see more broadly in the literature is that some confusion over the relationship between rainfall and malaria transmission. In many areas, the more it rains, the more malaria. In other areas, the less malaria when it rains. And of course, it depends on where those mosquitoes breed. I mean, some Anopheles fluviatalis likes to breed in some fast-moving rivers, but that's unusual. More often than not, they like a small, stagnant body of water. Now, we went to Tanzania to do some of this early work, and as part of that, we came up with this conceptual model based on the valley we were working in, about the different types of water bodies that existed in the landscape. So we had these topographically driven saturated areas, classic top model type approach, where the water table was intersecting with the ground surface and these puddles were developing. But also you get these small depressions, Hortonian runoff, these small short-lived depressions that exist in the landscape. Others on the floodplain, when the seasonal flood wave comes and the negative floodplain relief collects the water and gets left behind. Some breeding sites are in the rivers themselves that dry up in the dry season. They become disconnected and they become great habitats. But when the water comes back again, when the rains come back, those rivers flush through. And then groundwater springs, okay, others that are not related to sort of the terrain, but these are coming up from underground, and these are persistent year-round breeding sites. And what we did with this, the first of our projects, was to try and sample these water bodies at a monthly resolution, but being hydrologically and geomorphologically informed in our sampling scheme. So we tried to get each of these water body types. This was in near Ifakara with the Ifakara Health Institute, the Kilombero Valley of Tanzania, which had a really super high malaria transmission rate. And we had 61 larval sampling points that we visited every month, and we knew exactly the formation mechanisms of these from a geomorphological point of view. And we thought, why has this not been done before? And you can see the picture in the bottom left is an example of why that hadn't been done before. It was an incredibly difficult thing to get to some of these sites during the wet season. Even the beast there struggled from time to time. And so we deliberately sampled different water body types, and we recorded the details of these water bodies, temperature, and took larval counts from dips. We also set up a hydrological network of rain gauges, stage gauges, which itself was quite challenging. I'm not even going to talk to you about the evaporation pan that we had out there. And then we put these mosquito larval samples for genomic DNA extraction using PCR methods and identifying the species that we were getting from our dips. Now, the main result from that project is in this slide, and in the top right you can see a bubble plot of the larvae that were sampled in each of those water bodies, and that's just for November. So imagine a dynamic map of that moving, those bubbles moving up and down the landscape through the dry and wet season. And what we tried to do is relate the overall larval counts to hydrological metrics like rainfall, river stage, groundwater levels. And there was nothing, there was no statistical relationships. But when you disaggregate it by water body type, which nobody had done in the past, you actually do start to see these relationships emerge. So, our topographic convergences, funny enough, there's a negative relationship with water table depth. So when it intercepts the surface, they become more productive larval sites. The floodplain was the same, but with a 1 month lag. So only when the flood has receded do these become suitable for breeding. Same with paleo channels, these bigger features on the floodplain, but with a slightly longer lag, because I guess they take longer to become isolated. Again, river channels, we see a decrease after the rainfall, but actually that one wasn't significant for the year. Mostly we were catching Arabiensis, this cow-loving mosquito, perhaps because of bednet campaigns out there. And yeah, what we generally found was that rain was very variable and it wasn't the most helpful variable to use. But overall, this was showing, also, I should say, the groundwater was the kind of the star performer, to use a better word. This definitely had the highest overall larval numbers. And I guess cattle were getting used to where these places were and would be visiting and providing nutrients, shall we say, to that locality as well. But taking this approach allows us to have an understanding of this spatio-temporal variation in malaria vector larvae sites, and also understanding malaria risk and how that might change through the landscape as the wet season progresses, as different habitat types click on and click off through the arrival of the wet season high water levels. And following on from this, a few years later, we found ourselves in Zambia. So we move our story east here to the Zambezi River on the Barotse floodplain. This is 10,000 kilometers squared, inundated every year with flood recession taking place around June, July time. So super wide floodplain, around 10 kilometers, tens of kilometers wide in places, but lots of tributaries and depressions in the landscape known as dambos that also tend to have water in them year round. So about 2,200 225,000 people live on that floodplain. And one of the advantages of being here is that there was good health facility data to try and take this understanding that we got from Tanzania and move it down through the malaria transmission cycle. And we had brilliant collaborators with the district medical officers and the Ministry of Health there. So it was a really good opportunity to take these ideas forward. You can see a picture there from one of the kickoff meetings, and if you sort of highlight the posters in the background, you know, we were really surprised to see how difficult the malaria environment was there. So this is malaria incidence in under fives, and you'll notice an increasing line up to 2016 on that graph. But perhaps the most shocking thing are the numbers. Okay, that last number is 791 cases of malaria incidence per 1,000. Okay, so it's 3/4. And successes across Zambia in particularly bednet campaigns they were still being rolled out in Western Province, but it wasn't being as successful. They weren't seeing a positive impact on this malaria incidence for some reason. So we arrived with the same idea to do a geographically extensive and geomorphologically informed larval survey, and to try and relate that to the dynamics of the flood wave coming through the Zambezi. Two issues arose. The first was trying to use remote sensing to get an idea of where was wet and when. Landsat wasn't good enough because there was so much vegetated water and it couldn't detect that. And the second thing is healthcare access. So when we came to talk about these malaria trends, they said it's our second biggest problem, with the biggest problem being access to healthcare being cut off during the wet season and understanding when that's going to happen. So we integrated that into the project, as you'll see. To overcome the mapping of water bodies using satellites, we adjusted to radar using a Sentinel-1 based system. So C-band radar, which was kind of limited to grasses, but it worked much better than the Landsat system. So you can see in the image there, this is typically what we're looking at. It looks like a field, but it's actually a body of water. You know, you're paddling or wading through that. It's so much deep grass there. And that works, the Sentinel-1 works up to about 20 centimeters of vegetation, and then you need to use L-band beyond that, which is something that we've moved towards recently. So you get an idea of being able to map the persistence of water bodies through the landscape, and look how much of that is inundated vegetation. So the green area in the graph in the bottom left is not just water bodies, but vegetated water bodies. So it's a huge part of what's going on in this floodplain. And it had a 92% accuracy when we compared it with ground validation, but realized that there's no predictive ability here. So this is only so useful for us. So to try and move beyond that, we developed a floodplain model using ListFlood FP to try and get that predictive ability and examine how future climate changes might impact the water body availability in the floodplain, particularly thinking about changes to the timing and magnitude of the annual flood peak. Which we were seeing, it's damaging sort of traditional practices. The Barotse floodplain has the Lotse people on there, and every year they practice transhumance. So there's a ceremony where they leave the floodplain to go to a higher village for the wet season. And the timing of that has become disrupted in recent years. So we got high resolution DEM, a digital elevation model, and we did bathymetric surveys of the Zambezi over 40 kilometers and used a channel subgrid model using data from the Zambian Water Resources Management Authority. Lots of detail here. I'd refer you to the paper really, but we tried to, we did implement infiltration and evaporation in the floodplain model. This is an example of the flood wave passing through. One thing to note though is that we observed that in maybe 2014, the World Bank restored a network of canals on this floodplain that had gone into disrepair. And actually, when you model the floodplain with and without those canals, it has a huge impact on the water depth of the floodplain, which is what you can see in the middle panel there. Some areas were actually getting more water because of the canals. The water was being shifted over there more efficiently. Other areas were drying because of the canal. So that might in turn have an impact on the malaria transmission. So again, what we saw was a change in these locations of larval habitat through the landscape, but this time we used DNA barcoding, so it's like a full genomic analysis, so we could understand exactly what mosquitoes we were dealing with. We saw that the floodplain inundation was clearly important for Anopheles breeding. So the floodplain is a grey area on the left of each of those panels. And we saw increased productivity when the river levels started to fall. So dry season are the left panels, wet season are the right panels. And we worked with the University of Barotseland. We sort of trained up large teams to go and do these larval samples. And so we got a really good geographically distributed map of where the productive breeding sites were. Now, each row of that is a different type of mosquito. And what we would expect is that the reds and the purples of Anopheles, Phonestus, and Gambi would dominate that picture. But as you can see, it's the yellows and the greens dominating. So the most common vector was Anopheles custani. This was not what we were expecting to find here. And these are kind of cooler floodplain water bodies that Anopheles custani was breeding in. This is really, really significant and important because Anopheles custani bites you during the day. It bites you outside. Okay, so when we're seeing the lack of progress with the bed net distribution, this is most likely the reason why. So we backed this up later with adult trapping data that confirmed that what we were finding was Anopheles custani were actually the primary vector here, which was not supposed to be the case. And we detected them by this sort of geographically extensive larval sampling. And then the final point on this to say that the next step is to look at malaria transmission, right? And here we can see a map on the left of dry season and on the right of wet season, just for one single year. So there'll be some variability with the inundation occurrence overlaid. Again, you can see these dambos coming up, these little patches of permanently wet places. These are disconnected water bodies, but of course the floodplain. But look, there's not much malaria being recorded in the floodplain. And part of the reason for that, if you look at the figure on the top right, this is healthcare access map, okay, for the peak wet season. And you can see, of course, that no one can get to these health centres without a boat, which is actually used from time to time. So we can't discount that. That's something that we're developing into it. But you really have to consider health facility access to understand the population denominators to work with malaria data. So it becomes much more complicated. Yeah, and then just to work looking at how the Zambezi will change in the future. This is almost impressed. Responses to some very kind reviews have gone in. So Beth and Rose, my PhD students, looking at how we might see the timing and the magnitude of the flood wave to change moving forward. And we're generally seeing a drying. So we can now try and incorporate that into our understanding of how it might impact malaria transmission in this area. So as a final slide then, I just want to think about how we can take this understanding and move forward with it. And I would argue that this is a really good time for this sort of work. Our hydrological data and methods are advancing really quickly. What we really want to do is to take the two levels of detail I've presented here today, the continental scale on the left, the detailed floodplain scale on the right, and find that intermediate scale. That's where you can really inform this targeting and tailoring of interventions at a national malaria control strategy level. And that's what we're trying to do now. And I think with remote sensing data improvements and our machine learning driven classifications of that, we can deliver that in part, but also new generations of hydrodynamic modeling, this rain on grid hydrodynamics, where we can get a better understanding of surface water movement through the whole landscape. I think this is a really important area of development. And then we can try and embed that into a full malaria transmission model, not just looking at suitability, but actually thinking about where people live and how that will interact with the malaria transmission cycle as well. So this is stuff I'm trying to get off the ground. Let's just say I'm in between funded projects on this at the minute, but hopefully we'll change that situation soon. And then the final point is that this isn't just Africa, right? This is going to change around the world. And what I've been tinkering with is just taking that model that you saw earlier on, applying it to other continents, but for vivax malaria, okay? So not falciparum, just to see how Europe, how North America, South America, Asia, etc., might also see a changing suitability for malaria. So that's it from me. Again, I've got a huge list of people to say thanks to, particularly shouting out Chris Thomas, Tom Willis, and Andy Hardy, who've done a lot of this work, but many others as well, not least Beth Mroz, my current PhD student, working on this as well. So thank you for your time. Sorry, I went on a bit longer than I thought.
No, thank you, Mark. That was absolutely fascinating, and it really just highlights the complexity in understanding and the level of detail we need to go into to develop that better understanding of malaria, and how it links really quite keenly to geomorphology and hydrology. We've got about 5 to 8 minutes for questions. You should be able to unmute yourself or turn your camera on. So I'll open the floor now if anyone has any questions.
While we're waiting, Hannah,
Yeah.
this is where I have to remember to announce that we're hosting the BSG annual conference in Leeds next year. And the key dates for your diary are the 16th to 18th of September. So more on that soon. But if I didn't say that, I would get in trouble.
Yep, book it in your diary now, everyone. Helen, you got your hand up.
Hi, I'll just move you to the other screen. Yeah, really interesting talk. It really makes it clear how geomorphology is important and how you can use it to actually understand things that have such big impacts on people's lives, as opposed to just liking to go out and look at pretty rivers. But I just had a question about the future of malaria, I guess, because I think some stuff was saying that it was decreasing, but just thinking about it coming, moving north and south and away from those equatorial areas. Do we think that's a massive issue, or are we just so good at getting rid of it in some of these areas because we've already drained all our wetlands that it's not? I don't know. Any comment on that?
Yeah, I mean, there's a lot to say on that, really. I mean, you know, we've been able to get rid of it in Europe, especially because we were always on the fringes. And I think it's going to be harder for the malaria transmission cycle to restart from nothing. So it is going to be hard for that to come in. And I wouldn't be too worried about endemic malaria transmission in Europe, for example, for the time being. But you know, we're also talking about a different type of malaria with those final maps. That the Vivax malaria is not as lethal too. But what we're seeing is outbreaks like airport malaria, you call it. So, you know, someone's been traveling, they come back, and then it might start the cycle. And we actually have these Anopheles vectors that are capable of transmitting malaria through Europe. For example, I'm just using Europe as an example here. And what happens is now when we get these airport cases, there can be a few follow-on cases that are starting to happen. So I'm not saying this is becoming endemic, but these epidemics, these flare-ups might happen more and more and become more and more pronounced. We're seeing it in North America, like Florida, Texas. And then the other point to make on that, which I should have said in the talk, is that there are studies looking at not just malaria, but if malaria shifts out, dengue can shift in. So it's not just great for malaria. There's more things going on. And I really don't want to ever sell it as a good news story, because it really isn't. You know, there's more people living in there anyway.
We were in the Cook Islands just after they had, I guess, an airport breakout of dengue, and they were basically like spraying loads and, you know, doing a whole lot of stuff to try and stop it from going into the population. Yeah, it's quite a change.
Yeah, I think the change is most likely to be problematic, especially given that you build up this immunity. So
yeah, of course,
be a problem.
Cool, thank you.
Nice to see you.
Nice to see you too. Good talk.
Do we have any other questions? I've just got one. Yeah, if anyone's still thinking of some. I was just wondering have you been able to kind of disseminate this understanding to people on the ground in these countries and things, and what's been the kind of feedback and reaction to that?
Yes, sorry, I didn't really follow through the train of what happened with the Barotse example. So this is for Zambia. You know, we were very lucky to be working with the Ministry of Health quite closely, and we found out a lot about this a few years afterwards, just through a conversation over a meal, where we mentioned about these vectors, and at first it was thought that we were wrong, and then they did a follow-up study somewhere else, found the same thing, and then through that the Ministry of Health adjusted the intervention that was taking place in the Barotse floodplain in response to those findings. So it's kind of difficult to follow the chain through from one thing to another, but we have those links, and you know the projects were designed with the local district health offices where we work with them daily. One of them's doing a PhD on this sort of stuff. So it's completely wrapped up with them. That's the whole point, really.
Yeah, fantastic.
Good
That's
question.
really,
Thanks for that.
yeah, no, it's really good to hear. Do we have any other? Oh, we've got a question in the chat. Let me just get the chat up. Can improved accuracy in predicting suitable conditions for malaria vectors influence decision makers to invest in infrastructure projects that support malaria eradication efforts?
I guess so. I suppose, you know, a lot of it depends but infrastructure projects. Not sure quite what that means, but certainly health access would be one of the areas where infrastructure can help. And yeah, that's really important. But also, it doesn't have to be a big infrastructure project for that. You know, availability of ox carts and canoes and whatnot. You know, a big part of what we've been doing most recently has been working with a group of Canadians. I forget what it's called, like the Canadian Health Research. The funder there funded a project that we were partners on, and it was actually interviewing people and also the health offices about what they need, what the problems are, and it goes way beyond infrastructure. You know, this was more about maternal health than malaria, but because it's more geographically evenly spread, so you've got an idea as to you know the demand's going to be everywhere and how they get to the health offices, and people trying to avoid being taken to the health post, which would give a much better medical outcome for the births, but you know, they'd be taken long distances and there's no way getting home. So you know, there's other factors in there that we need to think about, especially for that, but also malaria as well.
Brilliant.
The canal stuff. I suppose maybe I think I'm not sure about. I think that we're a long way off that evidence chain about the reinstating the canals. That was nothing to do with malaria. It was more about the access issues, really.
Brilliant. Thank you, Mark. I'm just conscious of time, so I think we will wrap it up there. But thank you ever so much for that. Yeah, really fascinating talk on your research, and I look forward to hearing how it goes and develops, and hopefully there'll be a funding round soon that you can progress further. And it's such an important area of research. So thank you. We'll close the seminar there. So thank you, everyone.
Thanks, Hannah.